Crosses. When the flowering season arrives, collect the flowers of the plants that you will use as male progenitors. For this purpose, collect completely open flowers with the anthers near dehiscence or completely dehiscent. You can recognize this by the light brown color of the apical pore. Afterwards, separate the flowers from the plant by the peduncle and place them in a piece of parchment paper or craft paper bag, correctly identified with the progenitor's number or code. Then, take these flower samples to the laboratory. Disinfect your hands with an alcohol swab before extracting the anthers one by one with the help of tweezers. You can also carry out this same procedure by hand. In order to do this, first, remove the pistil and place the cone of anthers on a piece of parchment paper. Then, fold it to one side to keep the anthers inside and fold the ends to close it and prevent the pollen from falling during handling. After that, put the envelopes that contain the anthers to dry for 24 hours on a table with 100-watt incandescent bulb installed, keeping a distance of 48 centimeters between the light bulbs and the table. The aim is to maintain an optimum temperature of 25 degrees Celsius. 24 hours later, extract the pollen. For this purpose, first, disinfect your hands and the equipment you will use with alcohol. Then, shake the anthers vigorously, holding a vibrator against the external part of the envelope to help release the pollen. After that, open the envelope carefully. Remove the anthers, gather the pollen, and distribute it into one or more gelatin capsules, properly identified using a fine point indelible marker. Following that, close the capsule tightly and tap it so that the pollen drops to its bottom. Last, place the capsule in a wooden container specially made for storing several capsules in an upright position. Finally, disinfect your hands, vibrator, and table once again before extracting the pollen from another anther sample. For storage, prepare a plastic container with silica gel, a drying agent that prevents the deterioration of pollen caused by moisture. In order to do this, place the wooden container with the capsules on the silica gel. Otherwise, if such a container is not available, the capsules can be directly placed on the silica gel. Then, close and seal the plastic container with a piece of plastic sheet of extensible paraffin wax or parafilm wax, long enough so that it can be wrapped around several times to keep the container hermetically sealed. Finally, identify the plastic container and store it at 4 degrees Celsius, provided that the pollen is intended to be used within a maximum period of up to 2 weeks. Emasculation of the plants to be used as female progenitors. The flowers of the plants to be used as female progenitors are prepared for pollination during the early morning hours from 7 to 10 a.m. Begin this procedure by selecting an inflorescence that contains 5 to 6 flower buds of uniform size near their opening. Then, remove all the open flowers and very small flower buds that have not been selected. Continue with the emasculation, the removal of the anthers of flower buds, to prevent self-fertilization. In order to achieve this, open each of the flower buds with tweezers or a stylus and remove the anthers carefully to avoid damaging the pistil. Thus, the flower buds will be ready for pollination the next day. Pollination. The next morning, the emasculated flower buds will open their petals fully, exposing the pistil, which will be receptive to pollination. Carefully open the gelatin capsule from the male progenitor you will use in pollination, according to the crossing plan. Do this close enough to the pistil to allow the stigma to make contact with the pollen. Repeat this procedure with each of the flowers of the inflorescence. Then, put the crosses label around the stem of the inflorescence. The label should identify the progenitors used, starting with the number or code of the female progenitor, followed by an X, and the number or code of the male progenitor, the number of flowers pollinated, and the date of pollination. At this point, the formation and development of berries will begin. During this period, attach nets to the peduncle that holds the berries in order to avoid losses due to their fall when they ripen.